visi kami untuk menjadikan Universitas Bengkulu berkelas dunia pada tahun 2025.
Mengembangkan pendidikan dan penelitian merupakan misi utama kami. Kami tidak henti-hentinya mendorong seluruh sivitas akademika Universitas Bengkulu untuk mempublikasikan karya ilmiahnya di jurnal internasional bereputasi dan dalam bentuk hak atas kekayaan intelektual yang nantinya bisa diaplikasikan dalam pengabdian kepada masyarakat di skala lokal ataupun internasional.
Dengan tenaga pengajar berkualitas, terdiri dari 866 dosen dan 54 diantaranya adalah guru besar. Ini adalah bukti bahwa Universitas Bengkulu siap membentuk tenaga profesional yang berkarakter, berwawasan kemajuan, dan berdaya saing. Untuk menjawab tantangan perkembangan zaman, kami terus membangun fasilitas-fasilitas penunjang bagi mahasiswa, bukan saja untuk membentuk karakter, tapi juga sebagai wadah penelitian yang nantinya bisa bermanfaat untuk masyarakat luas.
Universitas Bengkulu adalah salah satu universitas yang penuh dengan kebinekaan. Berbagai macam suku bangsa ada di Universitas Bengkulu. Ada dari Papua sampai ke Aceh. Semua berbaur menjadi satu di Universitas Bengkulu. Universitas Bengkulu telah mendidik mahasiswanya dengan baik, sehingga memiliki kompetensi yang hebat, memiliki daya saing yang tinggi, bahkan telah berpartisipasi banyak dalam event-event nasional, event regional dan internasional, dan sudah banyak sekali prestasi yang ditorehkan oleh mahasiswa Universitas Bengkulu.
Untuk menunjang akademik, baik di sisi pendidikan, penelitian, dan pengabdian kepada masyarakat, kami sudah melakukan beberapa kerjasama seperti dengan instansi pemerintahan provinsi, instansi pemerintah kabupaten dan kota, perguruan tinggi negeri dan swasta, baik itu dalam dan luar negeri, serta dunia industri dan dunia usaha.
Ada begitu banyak yang sudah kami informasikan kepada Anda untuk bisa ditemukan di kampus ini. Saya mengajak Anda untuk terlibat dalam semua aspek orientasi, mempersiapkan diri Anda untuk belajar di Universitas Bengkulu sesuai dengan moto kami, menuju Unif Unggul Indonesia Hebat.
Saya Muhammad Fajri Hidayat selaku Ketua Laboratorium Kehutanan Universitas Bengkulu. Perlu kami informasikan bahwa Laboratorium Kehutanan Universitas Bengkulu itu sudah lama berdiri sejak tahun 1992, dan sampai saat ini sudah banyak melayani kegiatan akademik, penelitian, dan juga kegiatan pengabdian masyarakat yang dilakukan oleh sivitas akademika jurusan kehutanan dan juga Universitas Bengkulu pada umumnya. Oleh sebab itu, Laboratorium Kehutanan Universitas Bengkulu kami bertekad ke depannya untuk senantiasa berbenah diri, terutama dalam meningkatkan layanan prima laboratorium kepada mahasiswa, kepada dosen, kepada peneliti, dan juga kepada stakeholder lain yang berada di luar Universitas Bengkulu untuk melakukan kegiatan-kegiatan saintifik di Laboratorium Kehutanan. Oleh sebab itu, Laboratorium Kehutanan sudah menegaskan visinya, di mana visi daripada Laboratorium Kehutanan adalah terwujudnya layanan prima laboratorium untuk menunjang kegiatan akademik dan kegiatan pembangunan sektor kehutanan pada umumnya. Sesuai dengan tuntutan untuk menuju laboratorium yang profesional dan handal, maka Laboratorium Kehutanan saat ini memiliki 4 divisi. Yang pertama adalah divisi manajemen hutan yang dikoordinasi oleh Bapak Dr. Gunggung Seno Aji. Kemudian ada divisi ekologi konservasi yang dikoordinasi oleh Bapak Profesor Dr. Agus Susatya. Kemudian ada divisi silvikultur yang dikoordinasi oleh Bapak Insinyur Putranto Budiono Agung Nugroho. Dan terakhir adalah divisi pengolahan hasil hutan yang dikoordinasi oleh Bapak Profesor Dr. Ridwan Yahya MSc. Untuk mendukung layanan dan servis yang akan diberikan laboratorium kepada pihak luar dan kepada mahasiswa, maka kami memiliki 2 laboran yang saat ini senantiasa kami persiapkan menjadi laboran yang handal dan memiliki juga 1 tenaga administrasi yang digunakan untuk melayani kebutuhan administrasi laboratorium, baik itu oleh mahasiswa, dosen, dan juga stakeholder lain yang di luar yang akan menggunakan jasa laboratorium. Untuk mengimbangi kemajuan teknologi, ilmu pengetahuan dan teknologi di bidang kehutanan, maka kami selalu senantiasa berbenah untuk senantiasa meningkatkan layanan-layanan laboratorium yang bisa kami lakukan, termasuk juga analisis dan layanan jasa lainnya. Pada saat ini di Laboratorium Kehutanan kita bisa melakukan seperti misalnya adalah jasa identifikasi jenis tumbuhan, pohon, dan juga tumbuhan bawah. Kemudian juga kami juga bisa melakukan estimasi dan pengamatan satwa liar. Dan saat ini di Laboratorium Kehutanan sudah juga bisa melakukan layanan yang terkait dengan analisis tanah, seperti misalnya sifat fisika tanah, sifat kimia tanah, dan juga kami juga sudah bisa melakukan analisis karbon, sifat fisika kayu, sifat kimia kayu, pembuatan pulp, dan juga saat ini kami juga sudah memiliki studio analisis spasial, yang dimana studio tersebut adalah untuk membidangi jasa-jasa yang bisa kami berikan terkait dengan sistem informasi geografi. Jadi di Laboratorium Kehutanan saat ini sudah bisa melakukan analisis spasial yang juga cukup banyak, seperti misalnya pembuatan peta dasar, pembuatan peta tematik, kemudian pemodelan yang banyak digunakan di bidang di sektor kehutanan. Seperti misalnya kami sudah bisa membuat peta-peta tematik terkait dengan lahan kritis, juga pemodelan banjir, pemodelan longsor, kemudian dan pemodelan-pemodelan lainnya yang terkait dengan analisis spasial. Jadi dengan jasa layanan yang bisa diberikan, kami berharap bahwa ada dukungan dari semua pihak agar supaya Laboratorium Kehutanan itu kedepannya akan makin berkembang menjadi laboratorium yang memiliki layanan prima, profesional, dan handal. Saya kira demikian. Terima kasih.
kita kerja di sinar matahari. Gunung lembah berduri haruslah kita arungi dengan hati yang murni. Himbaraya, himbaraya, indah permainan mulia, matang tempat kita bekerja. Himbaraya, himbaraya, indah permainan mulia, matang tempat kita bekerja. Pagi petang, siang malam, tempat kita berseru. Bersatulah bersatu, tinggi rendah jadi satu, pertolongan selalu. Jauhkanlah sikap kami yang mementingkan diri. Ingatlah nusa bangsa, minta supaya dibela oleh kamu semua. Himbaraya, himbaraya, indah permainan mulia, matang tempat kita bekerja.
Dulu kehutanan, rimbawan, tetapi bingung di hutan, kemudian milih jadi botanis beliau. Today we have a talk about the ecology and evolution of Aristolochia. Itu seperti kantong semar. Saya belum pernah melihat itu. Tapi kita lihat, and then he will talk about the University of Dresden over there. It's a very good campus. And then in the end of the talk, he will offer some, if you interest on getting master in German, and you can apply to the university. Yeah, that's all. The time will be for Dr. Stephen. So, Dr. Stephen, you can start your talk.
Thanks a lot. Do I need the microphone? With or with the microphone? With the microphone? Okay, good. So, yes, welcome. I'm sorry for you waiting for a couple of hours. I'm really sorry for that. That's how it is. Sometimes electricity is down. Yeah. Okay. So before I start my scientific talk that is supposed to give you a tiny glimpse into the research that I'm doing, I'd like to introduce our university, so the place where I'm coming from. It's the Dresden University of Technology. Although it is a comprehensive university, it has a history in technology, but nowadays it is the only comprehensive university that has a strong focus on technology in Germany. For example, this here you see this is the biology building where I'm staying. My office is actually on the opposite side from this here, basically on that side. Is a very nice place. I will show you another picture later on. Okay, so I'm coming from a tiny country. I'm coming from Germany. Germany is there. Right now we are here somewhere. Germany is very small. And Dresden, the city where I'm from, where I'm teaching, is located here. You have Berlin here. You might have heard Berlin before as the capital of Germany. And Dresden is about 2 hours driving from Berlin. 200 kilometers roughly. And here you see the old part of Dresden with all the nice old buildings for which Dresden is well known. The Goldener Reiter and other monuments that we have. It's very nice to look at. So once you have the possibility to come to Germany, visit Dresden. It's full of tourists all the time. Okay, so a few facts about our university. I learned 2 days ago from your rector of the university that here you have about 28,000 students. Actually, in Dresden, we are not much bigger than that. We have roughly 33,000 students. And of those 33,000 students, roughly 5,000 students are international students from 125 nations, so pretty international. And we are enrolling every year about 8,000 students, roughly. We have 122 study programs from bachelor to master level and PhD. And we have many cooperations with university worldwide. Actually, I'm responsible for the international affairs of our faculty, and that's also one of the reasons why I'm giving a few slides about our university to communicate or to let you know that we exist, basically. We have roughly 8,000, approximately 8,000 employees, a little more than that, from which roughly half of them, are funded through third-party funds, so money that comes in from external sources. And in 2017, we had a budget of 530 million euros. I will give you another figure, of which roughly half of it, comes from third-party funding. And if you have a look at these figures here, you see that in 2007, We started with a third-party budget of 125 million euros. I don't know how to convert this into Indonesian rupiah. You have to do this yourself by your smartphone, I don't know. And within 10 years, we basically doubled this third-party funds that came into university. So we are one of the few universities in Germany that have this excellence status, and there's only a handful or two handful of those. Okay. And one of the very special things that we have in Dresden, and that is unique, is the so-called Dresden concept. And the Dresden, the name, is not only the city name, but it's also an acronym for Dresden Research and Education Synergies for the Development of Excellence and Novelty. And this is a unique combination with the four research societies that we have in Germany: Fraunhofer Society, Max Planck Society, Leibniz Association, and Helmholtz Association. Those are the four major scientific research societies that exist, and we have a very strong collaboration with all of those, and that's unique in Germany because these institutions are usually independent from university. And I'd like to point out the Senckenberg Word of Biodiversity, because I will come back to this very later on towards the end of my presentation. And with this, I'd like to jump to one of the research projects that we are doing that I'm very much interested in, and it's Aristolochia, the plant, the flower, one of the flowers you see here, and that's what we are talking about in the next couple minutes. We will talk about this plant, this flower, and this pollinator here. So we are talking about plants and flies, basically. Okay, Aristolochia, for those who are not so much into botany, they belong to a lineage that is called Piperales. That's the order. Piperales is the order and consists of a couple of genera that either have a parent or have no parent. So basically meaning the flower is nice, or the flower is super small and has no parent, and then you might need a scanning electron microscope to see the flower or any kind of magnification. Why are we studying Piperales? Well, it's the most species-rich lineage outside the monocots and eudicots, and it has the highest diversity in grow and life forms within those basal angiosperms, so outside the monocots and eudicots. They're extremely species-rich lineages, sister to extremely species-poor lineages, and this is very interesting from an evolutionary point of view. So Aristolochia, for example, has roughly 600 species. Here, Piper has about 2,000 species. Peperomia, another 1,600 species, whereas the lineage is Cipelia. You might know it because it also occurs in Indonesia. The genus Cipelia is only a single species in this genus. So one of the things that we are interested in is what makes some lineages species rich and what makes other lineages species poor. So here you see a couple of examples. So this is an example of Aristolochia from Central America. This here is called Hydnora, and that's basically the counterpart from Rafflesia, because it is also a holoparasitic plant. Hydnora is the name. You might want to Google it. And it's a fully heterotrophic plant, not making any photosynthesis, being leafless, and only producing the flower, similar to Rafflesia, basically. But it's not so big. It's just about this size, perhaps. So much smaller than Rafflesia. And other lineages here, like Lactoris, is also a single species that is shown here on this picture, is only occurring on a single island close to Chile, belonging to Chile, but 1,000 kilometers outside in the ocean. Okay, you might know pepper, right? Pepper, Piper nigrum. That's the pepper that we all use as spice every day, basically. It also belongs into Piperales. So we are talking here about a lot of economical importance as well. And yeah, so with this, I'd like to jump to the research to give you an overview about what kind of research I'm doing. So starting from morphology and anatomy, because it's fundamental to all the research that we are doing, you need to understand how a plant is built, what the morphology is, how the anatomy looks like, systematics and taxonomy, because you have to give the plants a name. So this is also very important. I'm very much interested in growth form evolution that also relates to plant grow. So why do a tree is a tree and why is the sister a liana? And how is this connected through biomechanical and water conductivity, for example? Comparative evolutionary genomics, interactions of pollinators, herbivory and parasite host interactions, biogeography and conservation, biochemical pathway and secondary metabolites that are also very important in these lineages, flower developmental genetics, speciation, diversification, and key innovations. And all this is connected through Piper Rayleighs. Although this is only one of the research that I'm doing, I'm also working in applied plant science and breeding science, for example. But today we're talking about basically about interactions and pollination and so on. And why am I interested in Aristolochia for the plant-insect interaction is because of two reasons. One is this side, that is the pollination. So you see the flower here, that is inside is hollow. It basically is a deceptive plant, and it collects the pollinators inside, in this case flies. And the floral structure, we will go more into detail in a few seconds, is covered by trichomes that you see here that are bending downwards and basically making a trap. I will come back to this in a few seconds. This is the pollination part. The other part, and that's why I'm here in Indonesia, is the herbivory, because these flowers here and the leaves are the host plants of birdwing butterflies. Birdwing butterflies look like this, and they exist here in Indonesia, from Papua to Sulawesi and Moluccas, Halmahera, and so on. So these butterflies are feeding on the host, on Aristolochia as a host plant. And that's why I'm very much interested in Aristolochia as well. We'll see if we get to this topic towards the end of my talk. So if you look at the diversity of Aristolochia plants throughout the world, here you see Aristolochia fimbriata or prostrata is coming from South America. This one here is from Papua, New Guinea actually. And this one here is from Africa, this one is from Central Africa here, and this one also from Latin America. And you see, although they all have different colors and a little bit of different shape, the functional morphology of this plant and of the flower is always the same. And but what I want to point out, it is a deceptive trap flower. So it is suggesting to give something to the pollinators, like nectar or other resources, but it's not. It's mimicking something else and is actually not offering anything to the pollinator, and that's why it's deceptive and it's a trap flower. Okay, and that's one of the reasons why I'm interested in this. If we look into publications that were published over the last 20 years, here's just a selection of it. Pollination in Aristolochia can be summarized as such. So what is generally thought about Aristolochia pollination? As we already said, they are deceptive trap flowers. They are fly pollinated. It's supposed to be brood site deception, so mimicking brood sites, breeding sites for the flies, and an order mediates the extraction of the pollinators. So this is the essence of what is published already before we did our research. So this is all from other authors before we started doing our research. However, at the point where we started our research, there was no evidence for models that are mimicked. In case that it is a deceptive pollination system, you need to have a model that is mimicked by the plant. And finding such a model is very difficult. So there is also very few detailed data on the pollination biology in general. And very often, and we talked about this already this morning with one of your professors, there is no distinction between flower visitors and pollinators very often in the published literature. So how do the people know if one fly is just accidentally trapped in a flower or is indeed acting as a pollinator? So this is a very crucial question. In general, fly systematics is super tricky. If you want to become an entomologist and you want to have a very challenging topic to work on with potentially a lot of merits that will be attached to your name in the future, if you do great research, then you might do fly systematics, because there are thousands and thousands and thousands of species that are still undescribed. Very often, we only know what is the male fly, and we have no idea what the female fly looks like. Yeah, so very often there are two species are described, one for the male and one for the female. On top of this, no broad-scale species-level fly phylogenetics are available. So nowadays you might know that we all use molecules, and we sequence the organisms, then we look up things in GeneBank and blast it against GeneBank. And the problem with this is that there are lots of flies are unidentified, others are just not included in phylogenies, so very limited knowledge about the phylogeny and molecular evidence from these organisms. And when it comes to fly biology remains largely unknown. Yeah, you know Drosophila, for example, Drosophila melanogaster or Drosophila suzuki. We know a lot about the biology, but about the millions of species that are outside of flies, we know very, very little. So, and that makes it very challenging when it comes to coupling fly biology and a pollination system. Okay, so let's have a look to the functional morphology of the flower, and that's very important to understand the remaining part of my talk, right? So if you look at the flower, although it might have a little bit different shape, as I showed you earlier, the functional morphology is always the same. You have a lip here, you have an entrance, this here is a tube, and of course this tube might be bended in different directions, we don't care for now. It is a tube, and this tube is covered inside with trichomes. And those trichomes, they are always pointing downwards. And additionally, those trichomes are covered by crystal waxes that prevent the flies to walk on it. So you know that flies have claws and so on, but with those crystals covering those trichomes, it's impossible to walk on these trichomes. So once a pollinator, a fly, is attracted and lands on the flower, it starts walking into this trap, and it cannot exit anymore. It's just walking downwards towards this part here that you see at this position, and here from the downs, from the below, it functions like a trap. And then inside here, this bloated structure, this is called an utricle, the flies are caught. The flies are caught in here, and what you see down here is a so-called gynostemium. This is the male and female part of the flower that is fused together to a structure that is called a gynostemium. So, what is happening during anthesis? So when the flower is opening, right? At the beginning, you have a female face of the flower, right? So at this stage, the gynostemium here looks like this. the anthers, these are the anthers, they are still closed, the pollen is inside, and the trichomes are turgescent, so they are fully hydrated, they are full with water, and they are functioning, right? And at that point, you see these trichomes here that can only push downwards, but you cannot pull them upwards because they would block at this position here, So an insect that is once in the trap cannot exit anymore. However, when it comes to the male phase, the flower morphology is completely changing. Here, the anthers are open, the pollen is shedded, and the trichomes, they shrivel. So within few hours, this is the same magnification. So here is fully turgescent, and here there is no water in the trichomes. And they look like this, and with this structure, the flies can start climbing on them. So it's actually a very clever thing that changes within 24 hours from this morphology to that morphology to ensure that pollinators are trapped, and then at a certain point, when they are covered with pollen, released from this trap. It's very sophisticated. So some of the research questions that we are doing, I will just read them. So what are the deceptive strategies of Aristolochia species in the Mediterranean? You know that I'm from Europe, so the closest area for me to work with Aristolochia is the Mediterranean. That's the area in the Mediterranean Sea in southern Europe, northern Africa. That's where I will show you a slide in a second where the Mediterranean is and where we are working, because it's very close to my home university. And particularly we are interested in the models that are mimicked. I told you that it's very sophisticated, potentially very sophisticated, and it's very difficult to investigate. And that's what we are interested. We are also looking into other questions such as, does the potential pollinator fauna differ among the study sites of the different Aristolochia species and or populations? Are differences in pollinator spectra based on differential pollinator attraction, or at least partly due to dipteran availability in the habitats of the different plant species and populations? I will not go into too many details of this, but just explain you how the research was developed and what was the outcome for a single species. So how do we study this? We have basically three work packages. The first work package addresses the pollinator fauna. So we want to know which flies are out there and which flies are attracted at the specific localities and also in the habitat of our plants. The second work package is the chemical ecology of the pollinator. So what is actually attracting our pollinators, and how is this done? And then we have the available Diptera community, so around the habitat of Aristolochia, to investigate if specific pollinators are just not attracted because they are not present, or because they can't smell the particularly odor that is elicited by the plant. So let's have a brief look into, I noticed that it's a little small, but I will help you reading it. So for this part, chemical ecology, we sample the scent. So what is the scent? If you have a rose, for example, you can smell it yourself. However, these are very few components that we can realize. Plants are eliciting a lot of different components, and Aristolochia is particularly complex. We are using the GCMS for the chemical composition and a method that is called electroantenography. I will show you in a second how this works. And with this, by coupling this, we investigate the behavioral attractive components in so-called bioassays. So we're going out with the chemical components into the field, and we are doing biotests in the field. And we are searching for the scent models at the end. On this part here, So we are looking for insects with pollen load so that we can ensure that if a particular pollen is found on the insect, we can investigate the pollen and say, okay, this is indeed Aristolochia. And with this, if we can find a pollinator in a female stage flower, then, or an insect that has a pollen load of Aristolochia in a female stage flower, then we can be sure that it was visiting another Aristolochia before, and with this is a very good indication that this is also a pollinator and not only a floral visitor, right? So, and finally, we investigate, we identify the species using morphological means as well as molecular means. And then we go out into the field and we use traps, all kinds of traps, to collect what is available in the insect community. We use barber traps, sweep netting and other things to collect what is available as insects, and then we use this in molecular data analysis as well. Okay, so today we will be talking about a single plant that is called Aristolochia rotunda, and we use this as an example for pollination biology in general in Aristolochia. Here you see a small map. This here is the Mediterranean Sea. Here this is Spain, this is Portugal, this is France, this is Italy. Here is Greece, this is Turkey, this is Northern Africa. And Aristolochia rotunda is occurring in this area that is labeled red here. And we are mostly working in Croatia, that was on this slide a second ago, here in green, along the coast here. And we are investigating 10 populations along the coast of Croatia between 2010 roughly and today. And down here you see the different habitats, from grassy areas of, where Aristolochia is growing. Here's the plant, the flower, in grassy areas, to stony habitats. So very different adaptations to the environment. And on this side here, you see the coast from Croatia, from the north to the south, with the localities with the blue dots that we investigate. So just a little bit more about the methods that we are using. So at the end, we are interested in the cues that are mediating the interaction between the flower and the insect, right, which is a fly. And we are using something that is called gas chromatography electroantennography, GCAEAD. How is this working? So we have a probe which contains the smell of the plant. The probe is injected here, and then is flows through these machines here, and you can identify the GC trace. So the individual components that are produced by the plant, they make these peaks here, and these peaks are standing for individual components, chemical components. And at the same time, here, down here, the chemical components are split into two directions. The first one goes into the GC measurements to make these peaks here. And the second goes into this here, into this part, and produces also peaks. But what you have here is the antennae of this insect. And we are measuring an electric signal of the antennae to know if a particular chemical component can be smelt or not, because only components that are that can be smelt can also be attracted for the fly species. And here, for example, you see that this component here is producing a signal in the antennae, whereas this component here is producing no signal. So with this, we can go to the next step, which will be field work, right? So we use these barber traps, these traps here to collect the insect in the environment, and also these simple bottles, plastic bottles that you all know, which we make holes in, and we can put in the chemical components and see if individual components are attracting specific fly species. And all these insects are collected and they go into molecular identification through barcoding approaches. Okay, so let's come back to this Aristolochia rotunda, the model or the modes of reproduction and pollination. So we did first some tests for autogamy, gnitogamy, and xenogamy that shows strict selfing can be excluded. But that's how it is. You can just be sitting and listening or sleeping, I don't know. But I need to be here, and to give you a nice talk. So we can say that strict selfing can be excluded. And of course, we need a lot of students that come in to do this research, because we are interested in who pollinates Aristolochia rotunda. And I will give you data from 7 of these 10 populations. So basically, most of the data that I'm showing comes from 7 of the 10 populations. We investigated 6,000 female flowers for visitors. Why female flowers? Because if we find visitors that have a pollen load in female stage flowers, then they have visited another Aristolochia before. So with this, we can be sure that they are indeed not only visitors, but also pollinators. From these 6,000 flowers, we found 1,100 arthropods being trapped and 306 carrying pollen. So of these 6,000, only 300 were left, because we can be absolutely sure that these are indeed pollinators. Everything else was left out. 88% belong to the fly family Chlorophidae, which is called halm flies. I will show you, there it is. This is one of these halm flies, and they are super tiny. They're about 1 millimeter in size, so super small. And of these 88%, there were again 84% belong to a single species, which we just called T. ruficeps in the future. And they were all females, with a very few exceptions. So you see how this looks like. This is a bag here full of flowers, for example, from this particular population. You have a student here that is making notes, and another student, and they're working in the evenings after field work in dissecting all the plants. And then they look for the pollinators. Here you see the pollen load on this fly, and here is a scanning electron microscopy picture of the pollen, and here a magnification of the pollen that is very typical for Aristolochia. So with this, we can be really sure. Test with individual components, so many different traps here, with acetone, with the synthetic mix that we used in the two-choice test, then with hexylbutyrate, with E2-hexanylbutyrate, heptylbutyrate, and so on, with a few others. And you see here on this side, the attracted chloropid flies. So we can see that the mixture here, it is very successful in attracting our flies, but also the HB and the E2HB is very attractive to the chloropid flies as well. So with this, we know that there are some components that are more attractive to the flies, to our pollinators, than others, right? But we have still no idea about the model that is mimicked. Yeah? What we know until this point is that we have female chloropid flies that are the pollinators, and mainly the species T. ruficeps. The flower scent is responsible for the attraction. That's what we proved positively in the biotests. we have highly complex scent composition, and major component classes are esters of fatty acids. And we see a high attractiveness, for example, for this HB hexylbutyrate. However, we have still no idea about what is the model. I'm recalling that this is a deceptive system. So if it is indeed a deceptive system, there needs to be a model that is mimicked. And this is very difficult to find out. Luckily, we found a manuscript that was published in 2004 in an environmental entomology, which is an entomological journal, by entomologists who are studying murid bugs that like this one here. And they said that their scent gland components of these murid bugs contain this HB and E2HB as major scent gland components. So this was very interesting because we see these components being produced by our plant. And they said that those components are attracting female chloropid flies. So they postulated a kleptoparasitic behavior of these chloropid flies. What means kleptoparasitic behavior? This means that let's assume you have in the environment, you have a predator, a spider or a praying mantid, like this one here. And this predator, is catching this bug. While this bug is eaten, those scent glands are destroyed. And when they are destroyed, they're eliciting these major components, which is called HB and 2HB, right? So this here is, this mantid or spider is eating this bug. The bug is releasing some scent, and this scent is attracting our flies. And exactly this scent composition is produced by Aristolochia fimbriata, by Aristolochia rotunda, to mimic this model, because this fly is interested in the hemolymphy of the insect that is eaten by this predator. It's super complex, but this was the hypothesis that we wanted to test. And so the hypothesis to be tested are flies attracted to the order of bugs being eaten, and is this order mimicked by Aristolochia rotunda? Super complex because it consists of different organisms and different degrees of dependencies. So to do this, we used a website that is called Pharobase, that contains all kinds of chemical components from nature. And we looked for chemical substances reported for the bug family Muridae, and we found 53 components being listed in this database. Then we narrowed these 53 down to chemical components co-occurring in Aristolochia flower scent. That is what we investigated before, and we narrowed this down to 22 components. Then we did GCEAD to see if those components can be smelled by our pollinators, and there were 7 components left. And then we determined the relative composition in the floral scent of these 7 components and used this in biotests using a new synthetic mixture containing chemical components according to this list here and this composition in this mix. These are details that are not so important. So the first sub-hypothesis that we wanted to test is this new synthetic mixture attracts our pollinators. Again, we use a simple 2-choice test in our field bioassay. And here we have the acetone and versus the synthetic new mixture that we have. And we have 0 versus 27 potential pollinators being attracted. So we can say, yes, it is attracting our flies. The second sub-hypothesis that we need to test is the model is existing on the same locality as the pollination system. because we need to prove that this exists. Otherwise, how do you want to mimic it, you know? So what we did, we went out into the field with entomologists, and they were collecting all kinds of insects that they found, and we squeezed those insects with a forceps to basically to kill them and to attract with those insects, try to attract our pollinators. And that's what is the result. Here you see one of these murrid bugs that was squeezed with a forceps so that the hemolymphy comes out and the scent glands are destroyed, and they're attracting those chloropid flies that you see here on the pollinator. And this was true for two different murrid bug species that are called Capsus arta and Notospira elongata, both murrid bugs. So with this, we were proving that the model that is the potential model exists on these localities. Another hypothesis that we needed to test to prove the whole thing is that attractive substances and components elicited by bugs are identical to those of our plant Aristolochia rotunda. So to do this, we used again GCMS analyses. So gas chromatography coupled with mass spectrometry. And what you see here is what is the bug, the scent components of the bug, and what you see down here is the scent component of the flower. So we see exactly the same peaks here for this E2 hexenylbutyrate and hexylbutyrate here and there. So we can prove that the scent is, the components are exactly the same, just the composition or the quantity is different between the murrid bug and what is produced by the flower. So with this, we basically discovered an entirely new pollination system that is a deceptive pollination system. However, so this was pretty cool. However, it is not the full story, because what I showed you here is just 84% of the pollinators, right? So at the beginning I was leaving out a few, let's say, unimportant things. But of course, we looked into this a little more in detail, and this here is the community of flies that we discovered in the 6,000 something flowers, and you see that this T. ruficeps here is not a single species, but it is 3 species. There is one here, and one here. Nobody knew about that before, but there are clearly 3 individual species that are mixed together in systematics, right? But this here is 84%, and this here is 16%. So until now, we did not look into this part here at all. But first of all, we also saw these 3 different potential 3 different species there. And if you link this to the localities where we did our experiments, then you see that some of the T. ruficeps here are found on many localities, but not on all. Those 2, for example, are not in these 3 triangles of the T. ruficeps biodiversity. So there seems to be a difference between individual localities that are super close to each other, and if you add the other localities to it, you will see that there might be a pollinator shift even in specific localities that are potentially down here, for which we need to have more data to see what are the details of the pollination system. We have understood how it is working in general, but there is still a lot more to do to understand the pollination system in total. So since we published this study a couple years ago, many other researchers were looking into this, and they found this same kleptomyophilic pollination system in other lineages. If you look at the tree of angiosperms here, Then you have the Piperales up here in the Mongolian clades. But meanwhile, they were also found, this same pollination system was also found in orchids and here in Ceropegia. So it seemed to be more widespread, this pollination system, but it always works in the same way. So to round this part up, as a take-home message for you, in contrast to many known examples of cheating angiosperm flowers, the pollination system described here has two novel aspects. The first one is that the plant mimic components released from freshly killed insects rather than living insects, and this is what was known before. And second, the flies deceived are not in search of food, which was postulated before, because it was said to be brood-side deception, but this is not the case because there is no brood-side deception at all involved in this pollination system. Because they look for food for themselves, they are looking for the hemolymphi to feed on them. So this new pollination system has been called kleptomyophilia. It is likely more widespread than we know so far, also known more further than the two other examples that I showed to you on the slide before. So with this, I'd like to end, and I think I still have a couple minutes. I'd like to end this part on the pollination and at least want to give you a glimpse into the other topic that we are interested in, the herbivore interaction and evolution between our plant here, the Aristolochias, and in this case here, this is a species from New Guinea, and the butterfly diversity that you see here. These are called birdwing butterflies. Many of those butterflies are protected in Indonesia, and they need Aristolochia as their host plant. The host plant is this one here, or any other Aristolochia. And the host plant specificity is super high because those caterpillars, they can only feed on one or two species of Aristolochia. If you put them on the different species, they will die. So it has also a lot of conservational implications, right? If you look to the diversification of Aristolochia that is shown here in red, in green, and the one from the butterflies, you will see that they occur on our Earth more or less here around 55 million years ago. This more or less 55 million years ago. They both occur on our Earth. That's according to our analysis that we did. And an ancestral area reconstruction shows that they were occurring in the northern hemisphere. That's how the Earth looked like 55 million years ago. And the reconstruction tells us that it was in the northern hemisphere, whereas now both the Aristolochia and the butterflies are most diverse in the southern hemisphere, or at least around the equator in tropical and subtropical habitats. So if we reconstruct a phylogeny of the butterflies, and this is the phylogeny of the butterflies that are using Aristolochia as a host and also relative butterflies, and we are mapping in colors the host plants. So in green we have Aristolochia, in pinkish we have Rutaceae, we have Annonaceae, Papaveraceae, Lauraceae, and so on and so on. And you go back to the center here, you see that the origin of everything was Aristolochia. So very likely Aristolochiaceae is the ancestral host plant of the butterfly on which they started their evolution. So the co-evolution started with Aristolochia and the butterflies. So if we look into the host plant shifts, because there are many different host plant shifts from here, from green to red, which means Zygophyllaceae, then from here to different blue, dark blue, Apiaceae, Crassulaceae, Saxifragaceae, and so on and so on. So many host plant shifts are visible during the evolution, and you map the net diversification rate. So how diverse are particular lineages of butterflies after the shift. So you can calculate this, and then you will see that with every single shift from one host plant to the next host plant and so on, you have an increase. Very often you have an increase in diversification. So with every shift, more diversification, more species, and this is why we see the diversification, the diversity of butterflies on our Earth, because they had so many shifts of host plants. And with every shift, you have new chemical components that they are using to defend against birds, predators, and so on and so on. So this is just a very brief overview about the research that we are doing otherwise than the pollination thing. Just a few slides as some teasers, right? And I would really like to have a few more students from Indonesia being interested in Aristolochia and the butterflies, because this is the diversity that you have in your country. I can't do this research because I'm too far away. I need to fly 15 hours to come here, you know? And it actually is also too hot for me. But you could do this research, and I'm super happy to support you in this research. So talk to your supervisors. Okay. So with this, if you think that this kind of research, the stuff that I was just showing to you, could be of interest for you, or not only to you, but to friends and colleagues of you, or you might say that this statement here is also something that you think is very important. The ongoing loss of biodiversity is one of the greatest challenges facing humanity today. If you think that this statement is important and you want to do something against it, or you want to study biodiversity a little more in detail, then you might consider a master that we are offering that is called Organismic and Molecular Biodiversity. And it's a joint venture between our university and Senckenberg World of Biodiversity, together with the International High School International University building in Zittau. So what is this? What is so unique about this master program? It teaches the fundamentals of biodiversity, its identification, documentation, evolutionary and molecular aspects of organizational levels, of 3 different organizational levels. The first one is the genetic variability within a species, so on population level. Diversity of species, the biodiversity itself, so how many species do we have, which species do we have, and so on and so on. And the ecosystem diversity, so bringing into these 3 individual levels of organization into one master. And we are unique in Germany because of the experience of the degree program. Why is this now so dark? Perfect. So this year Saxony is not very big. It's smaller than Sumatra, so don't care. And we have different localities here in where we do the teaching, because this is the knowledge of the experts that are involved in this master program. And we are merging people that are doing morphology, ecology, genetics, genomics, as well as applied topics that are related to biodiversity, taxonomy, systematics, collection management, and conservation into a single master. So you have all kinds of expertise coming together to develop new biodiversity researchers. So the master itself is organized in 4 consecutive semesters, and the semester starts in October. We are interested in students with an interest in biodiversity, natural history collections, and biodiversity research in general. What do you need to bring if you are interested, or one of your friends? You need to have a bachelor degree in biology or related fields. So you are studying forestry. forestry is something that we consider to be very related to biology. So it's actually much of the stuff that you are learning in forestry would also be taught in biology in our bachelor in biology. So we actually see this as very similar from the background, you know. So currently we have a very international team. So the teaching is all in English. There is no German teaching at all. So if you can more or less follow my talk, more or less, then you could certainly also participate at this master program. Currently, we have 17 nations from 4 continents being enrolled in this master, and this master started already only 2 years ago, so it's very new. And what we see as a possible career field for you to work in is, of course, natural history museums, research institutions, but also planning agencies, conservation biology and monitoring consultancies, governmental institutions, and NGOs that are somehow related to biodiversity. And many of the students that we have that do this master actually are coming from countries where they have a permanent position. They are working as lecturers already, and they get upgraded by doing this master, and then they go back to their home countries to teach this, what we have taught them, to teach this to their students. So it's a kind of multiplicator process, right? If we go into more details, as I said, it's 4 semesters. It starts with a leveling and basics semester, including some systematics of key taxa, statistics, applied ecology, basic molecular approaches in biodiversity research. That's actually the part that I'm teaching with lectures and practical classes. And then we have the 2nd and the 3rd semester, in which you have to specialize into different focal areas. You can study all 3, but you need to have one in which you collect more credit points. The first one is collection management. The second one is evolution of form and function, because it is essential to understand evolutionary biology, for example. And the third one is omics of life. So everything that is related to genomics, metabolomics, and so on and so on. And that's the part that I'm leading. And then at the 4th semester, you have the master thesis and your defense. If you are interested in this, you might either contact Carsten Wäscher from Senckenberg. He's the head of Senckenberg in Görlitz, or myself. There's my email address. And if you want to apply for this master, you can send a message to studium.ihi@CU-Dresden to Mrs. Scheunig. She's taking care of the administration. Or you just take a picture of this QR code that brings you to the website of our university, where you can look for more details of this master program. And with this, I say thank you, and I'm happy to answer questions. Don't be shy, I'm not biting.
Okay, wonderful talks. So first we have to applaud him. Stephen, I would like to summarize your talk in bahasa.
Yeah, go ahead.
Okay. Stefan bicara tentang interaksi antara spesies yang ada di Aristolochia dengan insek. Jenis ini seperti kantong semar ya. Kemudian dia punya bunganya itu punya corong. Kemudian corong itu berfungsi sebagai apa? Itu dicerangkan tadi. Kemudian juga antara insek dengan tumbuhan ini, mereka seperti komunikasi ya, seperti kamu menggunakan WA, kalau kamu dengan yayangnya, umpamanya. Oke. Hampir sama dengan begitu, tapi mereka caranya dengan chemical, dengan apa? Dengan senyawa. Kemudian karena bunga ini mengeluarkan bau, kemudian dianalisis biokimia yang ada di situ, kemudian juga serangganya juga dianalisis biokimianya. Ternyata dari itu ketemu bahwa mereka berkomunikasi dengan senyawa tertentu. Ya. Oleh karena itu yang tadi intinya diterangkan ke Stephen. Kemudian ada kupu-kupu dan evolusi dan seterusnya. Yang terakhir itu ada tawaran, apakah ada tawaran untuk master program yang topiknya adalah biodiversity, mulai dari ekologi kemudian evolusi. Jadi kalau kamu tertarik dengan itu, kamu bisa apply tadi di situ. Ya. Kita moga-moga ada 1-2 yang apply ya, apply dengan itu sehingga kita bisa. Kemudian kita waktunya tinggal berapa? 20 menit ya, Pak. 20 menit ya, Pak Agustin. Ya. Kita punya 20 menit untuk pertanyaan. Siapa yang bertanya silahkan. Kalau bahasa Inggris bagus, kalau bahasa Inggris, kalau pakai bahasa Inggris juga bagus, tapi kalau tidak nanti akan dicemarkan oleh Pak Jansen. Iya, yeah. Pak ya. Ya, saya pikir 3 pertanyaan dulu. Ada yang bertanya? 1, 2. Yeah. Yeah. We have to talk about that. Okay, it's okay. 1 kemudian 2. Jadi 2. The first part is there are 2 students will ask you something I do not know. It's going to be related to your topic. Okay, silahkan.
Baik, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you so much, Prof. Stefan Wankel, for the new insight. Because this is first time I know about the Aristolochia. I have a question for you. What kind of the ecological function, I mean special ability from the Aristolochia to handle our environmental balance? Do you get what I mean?
I'm not 100% sure if I understood the question correctly.
The plan for ecology. Yeah, for the ecosystem, I think.
The ecological function of this plant is a very difficult question because we have 300,000 angiosperms out there, and I think for very few of them there is an ecological function known. So to be honest, I'm passing out on this question. I can't give you an answer to this. It's just the diversity that we have. The function, I don't know. It has an importance for the pollinators. It has an importance for, no, not really, because they're deceived, right? So I don't know. I cannot answer this question. I don't know. Sorry for that.
Thank you, sir.
Ya, thank you, sir. Terus, Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Yoga Ridho Ilahi. Nanti Pak Yansen tolong Pak terjemahkan, Pak. Jadi untuk tentang tanaman Aristolochia ini, seiring dari presentasi tadi ya, saya itu menemukan di Google itu dari jenis Aristolochia ini di China itu menjadi obat herbal ya. Di mana dalam konspirasinya itu dari obat herbal tersebut ternyata itu mengandung beberapa efek negatif seperti kanker ya. Yang saya mau tanyakan bagaimana tanggapan Pak Stephen terhadap konspirasi tersebut. Terima kasih.
Like people, for example, in China use Aristolochia for herbal medicines, but it was found out that the compound, the use of Aristolochia for medicines might be, bad for the health actually, because it could cause cancer. You know anything about the herbal? I mean, the use of medicinal for the Aristolochia, something like that.
Yes. Did you look this up online while we had this talk, right? Yes. So I mentioned very early on in my presentation that Aristolochia is full of secondary metabolites, so a lot of secondary components, right? And some of the components that are used by the butterflies, or most likely used by the butterflies, those Aristolochic acids and Aristolochia lactams, that's why they feed on Aristolochia, because they put the chemical components into the caterpillar body, and this is protecting them because it's super poisonous from being eaten by predators, right? So that's at least the hypothesis. And these components are also very bad for us. There have been a couple of studies out there in the last 20 years, because Aristolochic acids and Aristolochic lactams are carcinogenic, so producing cancer and causing nephropathy, so kidney failure. And these two things have been found out because in traditional Chinese medicine, for example, Aristolochia is used, but very often it is used accidentally because it is confused with other Liana species, so of different systematics. And because the local population, they don't know, they don't distinguish between this plant and the other plant, and then they are mixing it and they are sending through the internet this to other places. And we had a lot of cases in Belgium where women were buying slimming products that are containing these traditional Chinese medicine treatments for getting slimmer. You know, you see me, I don't care about slim, but some women, they took this and they died because of this. There were many reported cases in Belgium, for example, that who died because of taking Aristolochia in traditional Chinese medicine that contained Aristolochic acids. Yeah. And there are also places in Croatia and in the Balkan. There is a sickness that is called Balkan nephropathy. Is because the people there they don't have the harvesting system as we have in Germany. They are not spraying that much with pesticides and so on, and herbicides. And they have Aristolochia plants growing into their fields. And when they are harvesting, they are also harvesting the seeds of Aristolochia, and then they are baking their bread, and then they develop this kind of sickness that was called in 2009 Balkan nephropathy, which was published in PNAS at that time.
Tapi salah, bukan semestinya yang lain. Nah, oke. Maksudnya spesies yang lain yang diambil mestinya dipakai, tapi ditambah Aristolochia ya. Di Balkan, di Bosnia, Kroasia itu pernah dipanen Aristolochia tumbuh, tapi dimanen yang lain bercampur sama itu. Tapi, can I ask something? Yeah. Stephen, can I ask something? No. No, I mean Aristolochia. I mean, for Aristolochia, is the butterfly very important for the pollinations, or okay, not at all? Okay. Yeah, yeah, yeah. But I mean the butterflies, but it could eat other leaves, not only Aristolochia or only Aristolochia.
Some of the birdwing butterflies that you have here in Indonesia
Only
are eating a single species of Aristolochia, not even two. Super specific.
Next one. Yeah. I will continue the question from the Janssens. So, in terms of the tritrophic interaction between the plants and then the flies, or maybe the butterflies and the natural enemies. For instance, is parasitoid predator. It is any good relationship between the parasitoid or predator to the plants, or maybe to the butterfly or flies. The second one.
I did not really get the first question. Can you repeat the question again?
Is there a relationship between the natural enemies? Because I cannot see your explanation between the natural enemies.
The natural
Enemies of the insects.
of the bug.
Of the flies, butterflies.
The natural enemy of the butterfly. Okay.
Related to
So,
the plants.
yeah.
Yeah. So is this any information?
So as far as I know, the caterpillars are usually eaten by, for example, birds, right? So the birds are flying around and they look for caterpillars to eat them.
Yes.
Right. So, but many of these caterpillars have very shiny colors. They're reddish, yellowish, orange. So colors that are indicating I'm poisonous, I'm dangerous, and so on and so on. And on top of this, they are actually poisonous because they collect the aristolochic acids, so the components that are produced by the Aristolochia plant. These secondary metabolites are in the caterpillar, and that's preventing them from being eaten by birds. That's the hypothesis. For example,
the Aristolochia produce some fruits, maybe, that can use for bird to attract?
the birds?
Yes.
Why? Why should the birds be attracted?
Because they need the bird to save the plants from the butterfly attacks.
You mean the plant don't want the herbivore, the caterpillar. I don't think there is a connection there.
Okay, so it means we still lost of the information between the relationship between the herbivore attack the Aristolochia with the natural enemies. It means if the butterfly attack the plants, it means the plants will be extinct, will disappear.
No. Yes. Well, but this is a balancing thing, you know. This is not a strong argument, because in nature the thing will not being extinct, because it is if you have a predator that increases in population density, let's say, then at the same time the host plant might go down, right?
Yes.
But because of a limitation of food, then you will also see a decrease of caterpillars eating the plant. And at some point, there is a curve that goes up. That's in ecology is very typical. So that is what happens in nature. It's just the human impact, for example, that is taking out parts of it or changing the climate or whatever, that is influencing the stable ecological system, I would say.
So what is the direct defense from the plants to protect from the herbivore?
I don't think there is one. I don't think there is one because there might be, but we don't know. Yeah. So there is no study on that. Yeah. We don't know if some specific chemical metabolites that are existing in Aristolochia are preventing other herbivores to eat them. Yeah. So why not mammals or rodents eating the leaves? We don't know. No study about that, right?
Okay.
We only know about the link between the butterflies and the plant because of the butterflies being so important for trade.
Okay, nice topics. Thank you.
Tadi Pak Agustin bertanya secara mendalam tentang interaksi antara Aristolochia, kemudian butterfly, kupu-kupu, kemudian ada predator di atas, ya, yang makan kupu-kupu. Bagaimana interaksinya? Singkat cerita, ada keseimbangan populasi antara 3 tingkatan ini. Untuk yang lain belum banyak pelajari. Jadi Aristolochia ini mempunyai chemical compounds, kemudian diambil oleh kupu-kupu, kimia ini, senyawa kimia menjadi toksik bagi predator di atasnya. Nah, jadi itu, oleh karena itu beracun. Oleh karena itu tadi diserangkan juga chemical ini mempunyai potensi untuk menyebabkan kanker. Kira-kira begitu ceritanya. Ada lagi pertanyaan? Pak Sapri? Okay.
Thank you very much, Prof. Stefan. In terms of evolution that you already talked, from all over the world that you already explore about Aristolochia, what kind of evolution that you find clearly? I mean, might be in terms of morphological or the color or what, and the other things. And secondly, I want to ask, what happened to the insect or fly that come to the tribe Aristolochia? I mean, the insect will be died, or and then what's that? The plant take something from the insect, or what is what is happen in there? So, yep, inside the flower.
Inside the flower?
Yep.
There's actually a small window at the bottom of the flower. It looks like a window, so there is no color in the flower. So the sun or the light goes inside. So what the flies are doing, they're just running around in the utricle, in this bloated structure, and because they want to escape, you know. And they get a little bit of nectar, a tiny bit of nectar that is produced by some glands, so that they just survive by running around in the flower during the anthesis of the flower, because the flower needs to change from the female phase to the male phase. Then the pollen is shedded, then they are covered with pollen, and then they can climb out of the flower and fly to the next one.
Oh, okay.
Yeah. So this is what is happening inside the flower. What was the other question?
In terms of the evolution happened in Aristolochia, what is the clear, attribute that you think is already changed? Might be in terms of morphological of the Aristolochia, or what? Until now.
I don't know. I did not really understand the question, to be honest. You mean, yeah, let's say 55 millions of evolution. Is how do we know? I mean, we can only study what is available right now on our Earth, the living plants, right? We can also study herbarium specimens that might contain plants that are not existing on our Earth anymore, but they were collecting 200 years ago. Yes, but what happened before that? We don't know. So we can model things, we can reconstruct those things based on distributional data, based on a fossil record. But to be honest, there is not a single fossil plant that has a flower of Aristolochia. We have a few evidence for fossils from fossilized wood, fossilized other generative, no, not generative, like leaves and other stuff that look like Aristolochia, but no flower, for example. So we don't know when this evolved in time, this system. Yeah, the flower might have looked different before. Yeah, because if we look at the sister genus, which is called Tothea, we don't have this trap system. So it's a bit of guessing, you know. I don't know. I would like to give you a better answer to your question, though, but I don't. I can't.
For Aristolochia for having the butterfly. I mean, what exactly the incentive, the advantage for Aristolochia, you know, hosting the butterflies? I mean, the butterfly is not acting as the pollinator, so what exactly the incentive? I mean, the benefits.
We don't know.
Okay.
It's just outside the plants. All the plants that you see outside will have any kind of parasite, will have any kind of insect eating on them, and so on and so on. For the majority, we don't know anything. We know for the plants that are of economic value, there is an interest to study this in more detail. But for the majority of the diversity out there, there is no interest. No, I don't. I don't know, and I don't think there is one mutualism in this case.
Okay, thank you, Prof. Stefan.
Okay, actually the time is let me thank us to interact with Dr. Stefan and also Dr. Yayan. So give him a big applause for very good talk today. So jika ada apa mau bertanya lagi dan seterusnya habis ini bisa ngobrol dengan beliau habis ini karena beliau tidak Jumatan ya nanti bisa ngobrol atau apa ya yang belum ditanyakan adalah kemungkinan sekolah di sana ya. Dr. Stefan, we are really thank you for your coming today and hopefully in the near future we have some kind collaboration between you and us, and maybe two of the students will, I do not know, if interest on biodiversity, and then you can go to master degree and that school. And we have some like a certificate for you and Pak Yayan because you has very good discussion today. So Pak Yayan, come here. Yes, you have to, or I will kick you out. Come here. Okay, come here. Yeah, Yayan, Professor Stefan, come here. So no, you have go to this. It's okay, leave me. Oh yeah. Thank you Pak Jansen, Pak Edi. Terima kasih semua mahasiswa semua, kemudian tim IT yang bekerja sangat bagus. Oleh karena itu tepuk tangan pada semuanya. Bapak-bapak, ibu-ibu, kita ada makan siang hari ini di ruang rapat jurusan. Saya nggak tahu anak. Yeah. Tapi kalau ada yang mahasiswa yang tertarik ngobrol dengan Pak Yayat, statusnya jomblo ya Mas ya? Boleh ya? Foto bersama. Steven, we are going.